Okay, now let's look at the cell-mediated response, the immune response with this. We've gone through the humora, which is the B cells, and the process of those becoming activated. So now this is going to be cell-mediated, and it's associated with the T lymphocytes or T cells. So let's take a look at this on what we're doing here. Okay, so we're going to start off with your naive T cell, okay? And we know that it has a receptor on this one. So let me do a different receptor than what we've done on the previous ones. So let me do a different receptor. So that's just a different receptor, okay? At this point, that naive T cell is not able to bind with the antigen, okay? It has to be shown how to bind with it, okay? So, in other words, another cell that is presenting the antigen has to connect with that naive T cell. Specifically, that one is, let me do this color here, that is going to be a dendritic cell. Now, if you recall back under the innate system, dendritic can be nonspecific. It's an antigen presenting cell. So what it does is it kind of swims throughout your system and it comes across various things that it doesn't recognize. It will phagocytose them and then present one of their antigens on its membrane. And specifically, it presents it on an MHC2 receptor. So let me put an MHC2 receptor on this. Okay, so let's say that dendritic cell ran across this antigen. Okay, so let me, I'll tell you what, let me let you know what this one is because we're going to change it. So this is an MHC2 receptor on that one. And what happens is the receptor on the naive T cell is specific for this antigen, okay? So the dendritic cell, the naive T cell is kind of residing in your secondary lymphatic structure, such like a lymph node, okay? The dendritic cell floating around your system comes across something, eats it, works its way to your lymph system, and it will present this antigen to the naive T cell. Now the naive T cell, remember under positive and negative selection, the process is that naive T cell must be able to bind with the MHC receptors, type 1 and type 2, or class 1 and class 2. It has to bind with that. It also has to be able to bind with that antigen, okay? So this naive T cell will bind with this component. So let me just keep it with the red. So that will bind, and in this process, it will activate. We've got other chemicals that activate, interleukin and stuff. Don't worry about those. This will actually lead to its activation. So this is activating it. Okay, those are energy things, whatever. Okay, so that's activating it. And now we have, let me just draw these a little bit bigger, activated T. Activated T cell with this one. And what the activated T cell can do, it actually can differentiate or change into different types of cells. One of which will be, I don't even remember the color, but I think it was purple. One of which is a helper T. A helper T cell. And what the helper T cell does is this one can actually bind with or activate the B cell. So remember that previous video that we did, helper T's will activate a naive B cell. So this is going to be activate a B cell. Okay, and then remember the B cell then leads into plasma or memory. The other thing that it can do, let me just do a couple different colors here. It can go to a memory, what would the, yeah, let's just do memory first. It could do a memory cell, memory T cell. Just as in with the B cells, a memory cell isn't doing anything for the current infection. It is residing in your body for future exposures. So if you are exposed to that antigen a second time, your body is able to respond to it much faster. Okay, so that's going to be a response for further exposures. Another one we're looking at is going to be the killer T cells, okay, or cytotoxic. They go by either one of those names, okay? So killer T or cytotoxic. And what's interesting about those is these have receptors on them as well, okay, that will then attack things that it does not recognize. So if we recall, let me slide over here. If you can recall that there are cells out there that are non-specific as to what they phagocytize. So let's say a macrophage, a dendritic cell, something along that line. So let me do a completely different color here. Let's go, and let me just do a, shoot, I'm using all my colors here. Well, let's just go orange on this one. So here we've got a cell, a non-specific cell that has phagocytized something, and it is presenting, it's an antigen, it is presenting that on an MHC1 platform. So MHC1 platform, okay? And it is putting, let me just do a pink one out here. It is putting some antigen that it doesn't really know what it is. It's just saying, hey guys, look what I found, okay? They ate something and now they're presenting it to their buddies, okay? So what happens is this killer T comes around and the killer T recognizes that cell or recognizes that antigen, okay? And what this antigen, let me just keep it with red to be consistent. That's red. Okay, so what it's doing at that point is it binds with this and it realizes, okay, we've got a foreign antigen that needs to be destroyed. And what happens to this cell is it undergoes apoptosis or programmed cell death. So basically, this cell is, killing that cell, or that cell is initiating apoptosis and it's destroyed. Now, there's a couple other cells, T cells, regulatory cells, and so forth. I'm not going to be worried about that. That was on a few videos ago that you can go back and look at as well. But this one, I guess you could use X's for eyes, okay? This one will end up killing these cells because this antigen could end up infecting other things. So basically, it's a very bad case of indigestion, okay? So that's food poisoning to its max. So going through this again, basically we have the naive T cell with the receptor. You have a dendritic cell coming along, presenting an antigen on an MHC2 platform. Presenting it on an MHC platform. This will activate that naive T cell. So now we have an activated T cell. The activated T cell can end up being a helper T, which then goes to your B cell, activates one of the B cells. It can become a memory cell or a killer T or a cytotoxic T cell and then the cytotoxic will go around and kill these other antigens presenting on an MHC1. So these would have to be also non-self antigens, okay? Because remember, part of your selection process, your T cells have to be able to determine self from non-self. So that is the cell-mediated response. What I want you to do right now, before you watch the next video, is I want you to take the flow chart that we did for your humoral response. I want you to take this one and put them together to show how the two interrelate. Okay, so do that, and the next video I'll come back and we'll go through it. Thank you.